Hello everyone, myself Nerityajeet. Welcome back to my YouTube channel. I hope you all are doing great, guys. So today's in this video we are going to learn about glycolysis. Also, this is a second lecture of respiration plant chapter. So without wasting any time, let's get started. Glycolysis. So there are two terms in glycolysis, that is glyco. Glyco means sugar and lysis. Lysis means simply break down sugar, or here let's say glucose. So it is process in which partial breakdown of glucose takes place to extract energy. So let's get started. So glycolysis takes place in cytoplasm in all living organism. This process takes place in all living organism, whether it is aerobic or anaerobic. So glucose, glycolysis is a 10-step reaction. Oxygen present or absent. In both aerobic or anaerobic type, it is sorry for notification, guys. So glycolysis is a process. It is independent of oxygen, and it is a 10-step process in which ATP is extract, NADH and H plus, and H2O is also extract. So in glycolysis, glucose partial breakdown takes place to form pyruvic acid. Then this pyruvic acid transferred to matrix or in the mitochondria. In matrix, carbon dioxide and NADH extract by the process of link reaction to form acetyl coenzyme A. And this acetyl coenzyme A enters into Krebs cycle. So Krebs cycle, and in Krebs cycle, energy and some other molecules come out. So let's start it. First, glycolysis. Some important points: all NADH plus H plus and FADH2 enters into inner membrane of mitochondria and participate in process, that is electron transport chain, in presence of oxygen, where one NADH gives 3 ATP and one FADH2 form 2 ATP. Energy is direct by ATP and indirect by NADH and FADH2. So first process is glycolysis. So it is a universal process which takes place in all living organism, no matter it is aerobic or anaerobic. So firstly, their simple molecule is glucose, which have 6 carbon. Glucose oxidize in presence of hexokinase enzyme at the cost of 1 ATP and ADP plus inorganic phosphate. This inorganic phosphate now attaches to the sixth position of glucose molecule and form glucose 6 phosphate, which is a 6 carbon and 1 phosphate molecules. Now this glucose 6 phosphate molecules again react in presence of phosphohexoisomerase enzyme to form fructose 6 phosphate. So after forming glucose 6 phosphate, it isomerizes to form fructose 6 phosphate by the enzyme with the help of enzyme phosphohexoisomerase. Remember this word, isomerase. Then fructose 6 phosphate use 1 ATP in presence of enzyme phosphofructokinase and Mg2 plus. Remember, phosphofructokinase is a pacemaker enzyme. So fructose 6 phosphate use 1 ATP in presence of phosphofructokinase, and this inorganic phosphate attached to the first position of this molecule and form fructose 1,6 bisphosphate, which have 6 carbon and 2 phosphate in the molecule. Now, this molecule breaks in presence of aldolase enzyme, which is a fourth step of glycolysis, and form two molecules of 3 carbon each, that is glyceraldehyde 3 phosphate, which have 3 carbon and 1 phosphate, also known as triose phosphate or PGAL, and other molecule of dihydroxyacetone phosphate, which have 3 carbon and 1 phosphate. Now at fifth step, an enzyme comes out, that is isomerase. So both these molecules are interconvertible into one another. Now at sixth step, glyceraldehyde continues this reaction, and also dihydroxyacetone phosphate is converted into glyceraldehyde 3 phosphate to form two molecules of glyceraldehyde 3 phosphate. In presence of phosphoric acid, or two molecule of phosphoric acid, one for each molecule, for one for each glyceraldehyde 3 phosphate, and two molecule for two glyceraldehyde 3 phosphate, because another molecule is come out from dihydroxyacetone phosphate. So two molecule of glyceraldehyde 3 phosphate in presence of two phosphoric acid uses oxidize reduces 2 NADA plus 4H to 2 NADH, and in presence of enzyme glyceraldehyde 3 phosphate dehydrogenase. Remember, this is a sixth step of glycolysis. Now at this step, this molecule convert to two molecule of 1,3-bisphosphoglyceric acid or BPGA. In this molecule, 3 carbon and 2 molecule of this carbon. At seventh step, this molecule changes to 3-phosphoglyceric acid. 2 molecule of 3-phosphoglyceric acid makes 2 ATP. So in this molecule, that is 1,3-bisphosphoglyceric acid, from each molecule one phosphate is come out and join with ADP molecule and form ATP molecule. So at seventh step, two ATP molecule is formed. At eighth step, enzyme uses mutase, and here two molecule of 3-phosphoglyceric acid now becomes two molecule of 2-phosphoglyceric acid. The phosphorus position is changed from 3 to 2. And at ninth step, another enzyme is used, that is enolase, and here water is come out, that is two molecule of H2O, and form two molecule of phosphoenol pyruvate. At this step, at this molecule, phosphorus position is now at first position of carbon. At last step of glycolysis, the 10th enzyme or the 10th step, enzyme pyruvate kinase is used and form two molecule of pyruvic acid in which 3 carbon atoms and 0 phosphate. At this 10th step, 2 ADP form 2 ATP with 2 inorganic phosphate. So, so as we see here that glucose is partially oxidized in this process, in this reaction, that is glycolysis. Glucose here is partially oxidized to pyruvic acid, and this is a phase of energy consuming phase or glucose activating phase. At first at first and third step, total 2 ATP consume. Total 2 ATP consume, and this phase is two molecule by fifth step and 2 by phosphate in one molecule by addition of inorganic phosphate. So it is an energy producing phase or payoff phase. So take a screenshot of this glycolysis. If fructose enters in this process, if fructose enters, then it directly use 1 ATP in presence of hexokinase enzyme to form fructose 6 phosphate. So remember, at first and third step 2 ATP used, and at seventh and 10th step 4 ATP form, and one NADH molecule is found from glyceraldehyde 3 phosphate, or we can say 2 NADH molecule form at sixth step of glycolysis. So this is important. Let's revise it. So glucose is converted to glucose 6 phosphate at the cost of 1 ATP, and then glucose 6 phosphate to fructose 6 phosphate, and then fructose 6 phosphate converted to fructose 1,6 bisphosphate at the cost of 1 ATP. So in both first and third step 2 ATP is used. Now at fourth step, with the help of aldolase enzyme, two molecule form, that is glyceraldehyde 3 phosphate and dihydroxyacetone. At the fifth step, with the help of isomerase enzyme, these molecules are both interconvertible into one another. Then at sixth step, 2 NAD plus convert to 2 NADH and from 1,3 bisphosphoglyceric acid. Now seventh step, 1,3 bisphosphoglyceric acid releases 2 ATP and form 2 molecule of 3 phosphoglyceric acid. Then from 2 molecule of 3 phosphoglyceric acid from 2 molecule of 2 phosphoglyceric acid, then 2 molecule of phosphoenol pyruvate. At 10th step, pyruvate kinase enzyme helps in changing phosphoenol pyruvate to pyruvic acid, and here at 10th step 2 ATP is formed. So so total ATP total ATP form in glycolysis glycolysis or substrate level phosphorylation. So what is substrate level phosphorylation? So when ATP is formed directly from substrate by taking phosphate from it, uh, then this is a substrate level phosphorylation in which ATP is formed directly from substrate. So answer is 4. So 4 ATP form, 2 at 7th step and 2 at 10th step. Net gain of ATP, net gain is 2, because 2 ATP used at first and third step of glycolysis. So net gain of ATP is 2, and indirect ATP is from NADH. Indirect ATP is from NADH. Let's take a new page. So indirect ATP from NADH. In our further lecture, we discuss how this ATP is indirect. So from NADH, so one NADH molecule, as we know, one NADH molecule releases 3 ATP. So 2 NADH molecule releases 6 ATP. So there is total gain of 6 plus 4, that is 10 ATP from glycolysis, from which 2 ATP is consumed during first and third step. So net gain of through this whole process, net gain of ATP is, that is 8 ATP net gain. But this 8 ATP only gain when aerobic respiration takes place. In case of anaerobic respiration, these indirect ATP, these indirect ATP is not formed. In case of anaerobic respiration, these indirect ATP is not formed. So in anaerobic respiration, net gain of ATP is 2 in anaerobic respiration. Now let's read NCERT of this topic. Glycolysis. The term glycolysis has originated from Greek word glykos, means sugar, and lysis for splitting or breakdown. The scheme of glycolysis was given by Gustav Emden, Otto Meyerhoff, and J. Parnas, and that's why it is also referred as EMP pathway. Because it is given by Gustav Emden, Otto Meyerhoff, and J. Parnas, also referred as EMP pathway. In anaerobic organism, it is the only process in respiration. Glycolysis occur in cytoplasm of all the cell. So it only takes place in cytoplasm of the cell in all living organism. In this process, glucose undergoes partial oxidation. Remember this word, it is partial oxidation to form two molecules of pyruvic acid or pyruvate. In plants, this glucose is derived from sucrose. As you know, in plant, glucose is derived from sucrose. So from sucrose is break down with the help of enzyme invertase to form one molecule of glucose and one of fructose. So which is the end product of photosynthesis? So in plant, sucrose is the end product of photosynthesis. Remember this point, this is important. Sucrose is the end product of photosynthesis. Or from storage, carbohydrate. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of enzyme hexokinase. So this is the first step enzyme. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Then subsequent step of metabolism of glucose and fructose are same. The various steps of glycolysis are depicted in figure this step, in which glucose at the cost of 1 ATP from glucose 6-phosphate then isomerizes to fructose 6-phosphate, and fructose 6-phosphate at the cost of 1 ATP convert into fructose 1,6-bisphosphate. Then by enzyme aldolase, two molecule form, that is triose phosphate, that is glyceraldehyde 3-phosphate, or triose phosphate, that is dihydroxyacetone phosphate. At sixth step, uh, 2 NAD plus convert into 2 NADH and 2H plus and form two molecule of triose bisphosphate, that is 1,3-bisphosphoglyceric acid or BPGA. Then seventh step, 1 ATP 2 ATP form from two molecule and form triose 3-phosphoglyceric acid, and then 2 phosphoglycerate, then 2 phosphoenol pyruvate, then 2 molecule of pyruvic acid form. at the last step of glycolysis. So in glycolysis, a chain of 10 reactions under the control of different enzymes takes place to produce pyruvate from glucose. While studying the steps of glycolysis, please note that the steps at which utilization or synthesis of ATP, in this case NADH plus H plus, takes place. ATP is utilized at two steps. First, in the conversion of glucose into glucose 6-phosphate, and second in the conversion of fructose 6 phosphate to fructose 1,6 bis phosphate, that is uh, at the step first and third. The fructose 1,6 bis phosphate is split into dihydroxy acetone phosphate and 3 phosphoglyceraldehyde. We find that there is one step where NADH plus H plus is formed from NAD plus. At which step? At the sixth step of glycolysis. This is when 3-phosphoglyceraldehyde or PGAL is converted into 1,3 bis-phosphoglycerate. Two redox equivalent or a redox equivalent, or that is 2H plus. 2H plus, two redox equivalent are removed in the form of two hydrogen atoms from PgAl and transferred to a molecule of NAD plus and form NADH by help of 1H plus from 1 NADH, and by help of two equivalent or two hydrogen atoms from 2 NADH molecule. PgAl is oxidized and with inorganic phosphate to get converted into bis-phosphoglyceric acid. This conversion of BPGA to 3-phosphoglyceric acid is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during conversion of phosphoenol pyruvate to pyruvic acid. So that is at 10th step. So can we then calculate? So we also calculate ATP. Pyruvic acid is then the key product of glycolysis. So the key product of glycolysis is pyruvic acid. What is the metabolic fate of pyruvate? So we discuss metabolic fate in the next lecture of this chapter. So this depends on cellular need. So this fate of pyruvate is depend on the cellular need. There are three major ways in which different cells handle pyruvic acid. So there are three major ways. These are lactic acid fermentation and alcoholic fermentation. Both are anaerobic ways, and the third one is aerobic respiration. So three major ways in which this pyruvic acid is handled by cell. Fermentation takes place under anaerobic condition in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to carbon dioxide and H2O, however, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires oxygen supply. So for further breakdown, Krebs cycle and link reaction pathway takes place, which is only possible in presence of oxygen. So thank you very much for watching this video till the end. So if you like this video, don't forget to hit that like button. And if you have any doubt, related to glycolysis, uh, type in the comment box. I'll definitely answer your doubt. So thank you very much. Bye bye.